23.1 and 23.2. Okay, so Chemistry 102. This is Chapter 23, Fatty Acid Metabolism. We're going to take a look at Section 23.1 right now, which deals with lipid metabolism in animals. And we fall under that category, humans do. So triglycerides are emulsified into fat droplets in the intestine by bile salts from the gallbladder. And when it says intestine, it's referring to the small intestine. This is really where lipids start to get broken down. And if you think about emulsification, it says here the triglycerides are emulsified. Well, what is emulsification? It means breaking the fats down, not metabolizing them necessarily, but breaking them down from large globules into fat droplets, so into something that's smaller. And what happens when we take a glob, let's say a sphere, and convert it into a bunch of smaller spheres is that we increase the surface area in that process. Now it says here that the triglycerides are emulsified or broken down into fat droplets in the intestine by bile salts that come from the gallbladder. Now, what are these bile salts made out of? Or what is bile made out of? It's made out of a few things. It's made out of micelles of lecithin, and we're going to look at the structure of what a micelle of lecithin would look like. Cholesterol, protein, bile salts. So we'll actually look at what bile salts are. They're part of the bigger umbrella of bile, inorganic ions, and bile pigments. And what happens is that after we break them down into these fat droplets, pancreatic lipase catalyzes the hydrolysis of the triglycerides into monoglycerides and fatty acids. And that pancreatic lipase is aided by another enzyme called colipase, which enables it to bind to the triglycerides. And then what's the fate of those hydrolyzed triglycerides going to be? They're going to get absorbed by epithelial cells and reassembled into triglycerides. Then they're going to be combined with proteins to form chylomicrons, which are going to transmit the triglycerides to the bloodstream. So let's take a look at how all of this works. So it says once triglycerides are in the bloodstream, they are hydrolyzed again into glycerol and fatty acids, and they're taken up by cells, and their fate depends on the situation. If energy is required, then the triglycerides are going to get used. They're going to get converted into ATP via beta oxidation, and that's going to be covered in section 23.2. And if not required, if the energy is not required, they're going to get stored in adipocytes, which is basically a fat cell. And you can see the structure of an adipocyte is mostly a fat reservoir, right? We have a little nucleus and some mitochondria there, some cytoplasm as well. But you see that the lion's share of what a fat cell is really just fat, okay? Anyhow, let's move on from there. Now, I said that bile could be made from micelles of lecithin. We looked at micelles of fatty acid carboxylate salts earlier on when we looked at soaps, the action of a soap. And the same rationale applies here when we're talking about a micelle formed from lecithin. Now, if you don't remember the structure of lecithin, this is a generic structure of lecithin shown here. You can see we have the glycerol backbone right here, and then we have a fatty acyl group, another fatty acyl group, but then we have this part of the molecule here where we have a phosphate, and we have this derivative of ethanolamine attached to it. So that's the structure of lecithin, everything that I have circled in green here. Now, what's the take-home message? The take-home message is that lecithin has a polar part, right? All these little purple spheres here, that represents the polar head group of the lecithin molecule. That would represent, this part here, which is polar. But then we have these big R groups here, which are nonpolar. Those are just long hydrophobic tails. So what's going to happen is when they're in the aqueous environment of the cell, all these are water molecules here, and the polar portion of the lecithin is going to interact with the water, and all the hydrophobic tails are going to point towards the inside, and they're going to serve as a place for the fats, like triglycerides, to conglomerate, so to speak, right? Because they're going to be nonpolar as well, and they're going to interact with that core inside. And so that's what a micelle of lecithin is. Very similar to the micelle that we saw when we looked at the action of a soap. Now, bile salts, on the other hand, it said that bile is also made of bile salts. Well, bile salts are derivatives of cholesterol. If you think about the structure of cholesterol, you can see that cholate and canodioxycholate are both derivatives of cholesterol. They both have that A, B, C, D ring structure. And we talked about steroids. Bile salts are made in the liver, stored in the gallbladder, and are secreted upon stimulus into the duodenum. So when fat arrives in the duodenum, which is basically the start of the small intestine, that triggers a signal for these bile salts to be released. Okay, and what do you see here? Now it's kind of hard to tell, but if you look on the far right, you can see that these are both carboxylates, right? We have a polar group, okay, and we have another polar group here. So we do have a polar group, but a lot of the molecule is hydrophobic. And so the polar portion is going to interact with the aqueous environment, but the hydrophobic portion is going to interact with the lipids, with the fats. All right, now this is a picture that I just took from another textbook. I thought it was a pretty, simplified view of digestion, but we have, the lipids coming in from the stomach. And then what does that do? Those fat droplets, when they reach the duodenum, again, they send a signal for bile salts in micelles and cholesterol to be released. And then we get emulsification, right? So we go from something that's a big droplet like these ones here, and we break them down into something smaller. Then they can get acted upon by lipases and broken down, and then they end up in my cells. All right, so that's something I just took from another textbook. I thought it was poignant. Anyhow, so emulsification, a little more about that emulsification, and this is how it's represented in our textbook. She says, okay, well, you start it with a large fat globule, like is shown here. We've discussed lecithin. We've discussed bile salts. This is kind of her just representation of what those might look like. We have a hydrophilic portion of the lecithin here, the polar head group. She's saying here the part that's in blue and the bile salt, that's the hydrophilic portion. Then we have the hydrophobic portion of the lecithin. We have the hydrophobic portion of the bile salt. And so when the bile salt and the lecithin interact with that fat globule to emulsify it, we get the emulsification droplets. You can see that the polar groups are on the outside, aren't they? And the hydrophobic portions, whether it's the long tail here or the hydrophobic region of the bile salt, is pointing towards the inside or is in the inside. And again, that just serves to provide more surface area or a greater surface area for lipases to act upon. Emulsification droplets are acted upon by pancreatic lipase. It hydrolyzes the first and third fatty acids of a triglyceride. So you remember the generic structure of a triglyceride, right? We have the glycerol backbone here, and here it's showing 1, 2, 3 fatty acid tails like that. Well, this is kind of interesting in that it hydrolyzes only the first and the third fatty acid, leaves you with a monoglyceride and 2 free fatty acids left over. So on the far right-hand side here, it says emulsification droplets are acted upon by pancreatic lipase. Okay, and this is just representing the enzyme here, which hydrolyzes the first and third fatty acids from the TGCs or triglycerides, usually leaving the middle fatty acid. Now, if you find this a little bit difficult to follow, if you're like, what the heck is this again? Well, I have a bigger structure here. This shows the full-on structure of a triglyceride. And again, the pancreatic lipase is going to hydrolyze 1, 2, right? It's going to hydrolyze the first and the third ester bonds to produce 2 free fatty acids and the monoglyceride. So that's just, basically a blow up or maybe a clearer picture of what was on the last slide. Okay, so we've gotten that far. We've broken the TGC or the triglyceride down into a monoglyceride and free fatty acids. Well, then those are going to combine. So the monoglycerides, the free fatty acids, are going to combine with cholesterol and fat-soluble vitamins, and they're going to make new micelles. Okay, so it says several types of lipids are going to form micelles coated with bile salts. And this is where we really get to something that can start to be absorbed by the human body. And in order for it to be absorbed, they have to be packaged into chylomicrons. So intestinal cells are going to absorb the lipids from micelles. So we get the micelles being absorbed across, and it says here an absorptive cell, like an intestinal cell. The triglycerides are then going to be repackaged or reassembled, which is kind of surprising. And then they are packaged into protein-coated chylomicrons. You remember that we talked about chylomicrons earlier on in the class. So if we look down here at the bottom, it says intestinal cells absorb lipids from micelles, resynthesize the triglycerides, surprising, and package the triglycerides, cholesterol, and phospholipids into a chylomicron. Okay, so now we've got our chylomicron. It's not in the bloodstream yet, but the chylomicrons are absorbed into the bloodstream where they get hydrolyzed again. And that's kind of an interesting process. I don't really go into a lot of detail about this in my video because our textbook doesn't really go into a lot of detail about what the Golgi complex does. But it says here the Golgi complex packages those chylomicrons. Okay, so here are the chylomicrons right here, packages them into secretory vesicles. That's what's shown here. And then the chylomicrons are released from basal cell membrane by exocytosis and enter the lacteal and basically end up in the bloodstream. Okay, now that's kind of a simplified version of what happens, but it will certainly suffice. Now, what about the lipid storage? Well, once the fatty acids go into the bloodstream, they can be stored in adipocytes or adipocytes, however you pronounce it, as triglycerides in the cell's cytoplasm. And when you need energy, you're going to hydrolyze those triglycerides to fatty acids, and the fatty acids are going to get transported into the matrix of the mitochondria where they get oxidized. And again, that's section 23.2. And complete oxidation of fatty acids, which we call beta oxidation, so this is called beta oxidation, releases much more energy than a comparable amount of glycogen. So we can harness or recover a lot of energy from triglycerides in the process of beta oxidation. So let's take a look at that process that we call beta oxidation. Now, the title of section 23.2 is simply fatty acid degradation, the oxidation of fatty acids. And if we historically, if we look at the history of the study of beta oxidation, a chemist named Franz Knoop did a really interesting experiment a long time ago where he took phenyl-labeled, it says here, oxidation of phenyl-labeled amino acid occurs 2 carbons at a time. So this is a mistake. I just looked at this and I'm like, that's not right. It should say phenyl-labeled fatty acids. I'm not sure if you remember from Chemistry 101, you would have studied radio-labeled compounds where they put a radioactive element in a compound. Now that's something that we do, nowadays, but that wasn't known at the time that Franz Knoop did his experiment. But what he did is he took what he called these phenyl-labeled fatty acids. So basically a fatty acid that has a phenyl group at the end. Okay, we have a long hydrocarbon chain, and then we have a phenyl at the end. We call these omega phenyl-labeled fatty acids. Okay, so he took 2 different kinds. Okay, he took fatty acids that are omega phenyl-labeled fatty acids. He took some that had an even number of carbons and others that had an odd number of carbon atoms. And then he studied the products of the metabolism of these fatty acids, and he found that when the omega phenyl-labeled fatty acid had an even number of carbons, that he ended up with acetate and phenylacetate. However, when it had an odd number of carbons, he got acetate as well, but he got benzoate. His conclusion was, he said, okay, well, the degradation or the metabolism of these fatty acids must be occurring by 2 carbons at a time. And he said, okay, well, I'm just going to make, acetate. Now, nowadays, we know that acetate is in acetyl coenzyme A, but his conclusion was correct that we lose 2 carbons at a time. Now, that's just a little bit of history on discovering the pathway of the oxidation of fatty acids. But an overview of what we know nowadays is that fatty acids, yes, they are degraded into 2 carbon fragments in a process that we call beta oxidation. The first step is an activation step. Okay, so we have to activate our fatty acid. And then steps 2 to 5 are a set of 4 different reactions with a basic outline close to the last 4 reactions of the citric acid cycle. Each pass through the cycle releases acetyl coenzyme A, and since acetyl coenzyme A has two carbons, acetyl coenzyme A has two carbons in the acetyl group, it's going to return the fatty acid as a coenzyme A, but with two fewer carbons. So basically, we lose two carbons with each cycle of beta oxidation. Okay, and for every cycle of beta oxidation, we release one molecule of FADH2 and one molecule of NADH. Okay, all right. So not only do we produce acetyl coenzyme A, which can enter the Krebs cycle or the citric acid cycle, but we also produce these two coenzymes, which can be oxidized through oxidative phosphorylation and produce even more ATP. So again, you can see how we can harness a lot of energy here. Now here's an overview of the process of beta oxidation. And just like we saw with glycolysis, you can see that the first step actually costs us energy, doesn't it, right? We're starting with ATP and we're ending up with adenosine monophosphate, right, AMP. So this is the investment phase here. But you can see here that every step after that is nothing but a payoff phase. So we get a big payoff for our early investment, okay? In the second step, we produce an FADH2 molecule. We know that those are worth 2 ATPs. In the fourth step, we produce an NADH molecule. We know that those have a value of 3 ATPs in oxidative phosphorylation. And then finally, all those acetyl coenzyme A molecules, for every one that you produce, they're going to go into the citric acid cycle and they're going to produce 12 ATPs. And if you're wondering about counting up ATPs, we'll look at the last slide that we're going to look at in this slide deck. We'll deal with counting up ATPs, and I promise it won't be that bad. Okay, so let's take a look at the steps. It's just so you have an idea of what the heck is going on. Is it oxidation, reduction? What's going on in the beta oxidation process? Well, enzymes used in beta oxidation are located in the matrix space of the mitochondria. But we have our fats out in the cytoplasm, so we have to carry those into the matrix space. So we need special transport mechanisms that are required to move the fatty acids into the mitochondrial matrix. And in step 1, we do an activation reaction that produces a fatty acyl coenzyme A molecule. Let's take a look at that. So here's our fatty acid shown right here. This is our investment phase, and we end up with a fatty acyl coenzyme A. And you can see that this is a thioester right here, and this squiggle bond right here is a high energy bond. So a high energy thioester bond is formed between coenzyme A and the fatty acid, but the reaction requires ATP. Now, since we end up with a pyrophosphate here, this is like the investment of 2 ATPs. So it's really costing you the value of 2 ATPs, and you're going to see that when we tally everything up. The hydrolysis of the 2 phosphoanhydride bonds, so it's like the value of 2 ATPs or the cost of 2 ATPs. Energy invested at the start of the cycle is going to return a much greater amount of energy in the pathway. And this reaction is catalyzed by an enzyme called acyl coenzyme A ligase, which is located on the outer mitochondrial membrane. And the product is an activated acyl group. That's what we want to get, right? We have our high energy thioester bond. Well, let's look at a little more of the details here. How do they cross into the matrix? The activated acyl group is going to react with a molecule called carnitine, which is a carrier molecule. The reaction is catalyzed by an enzyme called carnitine acyltransferase 1. That's going to transfer that fatty acyl group to the carnitine molecule. We're going to look at its structure in a second, which is going to produce a molecule called acylcarnitine, and it's going to liberate coenzyme A. So you're like, well, you know, 1 step forward, 2 steps back, right? But hold on. The acylcarnitine is then going to cross into the mitochondrial matrix via a carrier protein called a translocase, located embedded in the mitochondrial inner membrane. And then the fatty acyl coenzyme A is going to be regenerated by a transesterification reaction catalyzed by a second enzyme called carnitine acyltransferase 2. So let's take a look at the pathway here. First let's take a look at the structure of carnitine. So here's the structure of carnitine. It's this molecule here. I don't ask you to memorize that structure, but what happens is it reacts with the fatty acyl coenzyme A here, okay, via this enzyme, carnitine acyltransferase 1, and then you produce an acylcarnitine, you re-liberate coenzyme A, and the reason this happens is so that this portion can be transported into the matrix, right? Otherwise, it can't happen. Now, this is the figure that I was looking forward to here. So here we have the cytoplasm, right? We have our fatty acids out here in our adipocyte. Okay, well, how do they get from the cytoplasm into the mitochondrial matrix? Well, first, the fatty acyl coenzyme A is going to react with carnitine via carnitine acyltransferase 1. So that's this enzyme here. We produce the acylcarnitine. We looked at the structure on the last slide. That is going to be able to go across the outer membrane and into the intermembrane space, then to the so this is the translocase here. So now you've got your acylcarnitine, it kind of like snuck into here, and then it's going to react with carnitine acyltransferase 2, which is going to re-liberate the acyl coenzyme A. Now, if you're wondering, why couldn't they have just designed it to go from here to here? Well, it doesn't work, right? It doesn't work that way. Okay, and then carnitine is going to come back out, and the process is going to repeat itself. Well, now we have what? We have our fatty acyl coenzyme A molecule inside the mitochondrial matrix, where the enzymes are for steps 2 to 5, the complete beta oxidation cycle. So let's take a look at that. This step 2, is an oxidation reaction. Remember, oxidation is gaining oxygens or losing hydrogens. This step is an oxidation reaction, removing a pair of hydrogens from the fatty acid. Okay, we're going to remove a hydrogen from this methylene and this methylene. Where are those hydrogens going to go? They're going to go to reduce FAD to FADH2, which again can enter oxidative phosphorylation and produce 2 ATPs. This dehydrogenation reaction is catalyzed by an enzyme called acyl coenzyme A dehydrogenase, results in a carbon-carbon double bond. During this process, again, I'm repeating myself here, but FAD is reduced to FADH2, and oxidative phosphorylation is going to give you 2 ATP molecules. Okay, so that's step number 2. So we've removed 2 hydrogens. Okay, great. Well, remember that this process is called beta oxidation. And now that this carbon is sp2 hybridized, now it's ready to be hydrated. So that's what's going to happen next. Is the third step is the hydration of the double bond, right? We're doing a hydration, right? We're going to add a hydrogen and a hydroxyl, right? We're adding water across this bond. Remember that it's called beta oxidation, right? Alpha, and this is the beta carbon here. So there's the oxidation, okay? So the hydroxylation is considered an oxidation, and this is catalyzed by an enzyme called enoyl coenzyme A hydrase. And then we're going to oxidize that further in a dehydrogenation reaction, where we're going to lose this hydrogen, that hydrogen rather, and this hydrogen to form a carbonyl. And now you can really see where we're going, right? So this oxidation reaction is catalyzed by L-beta hydroxyacyl coenzyme A dehydrogenase. I'll never ask you to remember that, okay? But you should know that in the third step, NAD plus is going to take one of those protons and it's going to get reduced to NADH. NADH, we know from chapter 21, when it undergoes oxidative phosphorylation, it's going to produce 3 ATPs. Now we have the final player, right, in the whole process. Because what's going to happen here is that coenzyme A is going to come in and it's going to react, and it's going to release a 2-carbon unit. So if we back up here, I just want to point this out to you here. Okay. Sorry, just let me double check that everything's correct here. Okay. So we're going to end up releasing these 2 carbons here. Just watch. Okay. You see that these 2 carbons here, you have your coenzyme A, which has a thiol on it, and this thiol ends up attacking right here. Okay, so that's what happens, and you end up breaking this bond and adding a hydrogen to it. So that's what we call a thiolysis, right? Because we have a thiol and it's a lysis, right? We're breaking this bond here in the process. Now look what happens. You see there's a lot of similarities here, right? This molecule and this molecule look the same, don't they? But they're different, right? This one says CH2 to the power, or N, and this one says CHN minus 2. So that means we've lost 2 carbons. And if you're wondering, well, can't that just happen over and over and over? And the answer is yes, that's the whole point. It keeps happening over and over. In addition to acetyl coenzyme A, that's here. Okay, that's acetyl coenzyme A. This is the fatty acyl coenzyme A, is 2 carbons shorter, and the whole process can start over again, right? That can go in again. So the shortened fatty acyl coenzyme A is further oxidized with additional cycles until the whole thing is broken apart or is degraded to acetyl coenzyme A. Each acetyl coenzyme A produced enters the citric acid cycle, right? Remember when we had the 2 pyruvates and we made 2 molecules of acetyl coenzyme A, and then we made 24 ATP molecules? Well, here we're only producing 1, so we're going to make 12 ATP molecules in the citric acid cycle. And there we go. So if we tally up the complete oxidation of palmitic acid, and this is just an example, okay, a very specific example, but it will help you to understand what the heck is going on here. And I don't know if I should be recording this, but I can tell you a question that was asked on a previous final exam. So if we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbons in palmitic acid, right? 16 total carbons. What I said in my video that I recorded and posted to YouTube is I said it might be a knee-jerk reaction to say, well, if I've got 16 carbons in palmitic acid, hey, I can break that up 8 times, right? But that's not true. Okay, you can break it up into 8 molecules of acetyl coenzyme A, but how many times does the beta oxidation cycle occur, right? 1, 2, 3, 4, 5, 6, 7. It only occurs 7 times. Okay, so it's kind of like you divide by 2 and then you subtract 1. Okay, so let's tally up how much ATP would we get from palmitic acid. Well, remember that the investment phase, right, cost us the equivalent of 2 ATPs. Okay, so once we get to that phase right here, now we've got our, palmitic acid coenzyme A derivative. It's called palmitoyl coenzyme A. That's neither here nor there. It's going to undergo beta oxidation how many times, right? 7 times. In 7 times, yes, we produce 8 acetyl coenzyme A's, but we've only undergone the process 7 times. So that means we're going to produce 7 FADH2s and 7 NADHs. Right? FADH2 has a value of 2 ATPs, so we produce 14 ATPs. NADH has a value of 3 ATPs, so 7 times 3 is 21. Okay, good. So we've got those 2 right there. Let's highlight them just so we can kind of keep track. I meant to highlight them in green. Where's my green highlighter? Okay, so far we got 14 plus 21 if you want to get a calculator out, right? 35. Okay, so what happens to these 8 molecules of acetyl coenzyme A? They can all go into the citric acid cycle, right? Okay, well, we know that in the citric acid cycle, we're going to produce 3 molecules of NADH. Okay, so we have 8 times 3, which gives us 24, but we know that each NADH has a value of 3 ATPs. So that gives us a total of 72. We produce 1 FADH2. Each one of those is worth 2 ATPs, so 8 times 2 gives you 16. You produce 1 GTP. Hey, that's just got the value of an ATP, so that gives you 8 ATP. Maybe I should put the number 8 here. So give me 1 second. So we produce 8 ATP there. I'll highlight it in green to keep track. Okay, so if you tally up, and I've got a calculator here, so if you take 35, so that's 14 and 21, plus 72, plus 16, plus 8, that equals 131. That's your grand total. But again, you invested 2 at the beginning, so you subtract 2, and you get a total of 129 ATPs. Now, what it says in our textbook is that you recover about 3.5 times more energy from a triglyceride, or from a fatty acid, I guess is what I should say, than you would from a comparable amount of glycogen.